Hello, everybody. So today we are jumping into dihybrid crosses. A lot of what we have done so far has kind of been building up to this and getting ready to basically set these up and solve them. So today's the day. We finally get to solve a dihybrid cross. What we're going to do is basically just go through an example problem. I'm going to ask you to periodically pause throughout the video. So when I do take a second, pause, and then try to figure out the answers to the questions that I'm going to be asking you. So to start off, we're going to do another example very similar to my last video about gamete formation. So we're still going to consider the two genes of seed color and seed shape, where green, big G, is going to be dominant to yellow, little g, and R, big R, round seeds, are going to be dominant to wrinkled seeds, or little r. So in this example, parent 1 is going to be heterozygous for seed color and heterozygous for seed shape. So my question to you is, what is the genotype of parent 1? Go ahead and quick pause the video. Please go ahead and figure out the genotype of parent 1 based on this information. They're heterozygous for seed color and heterozygous for seed shape. All right, so what you hopefully should have come to is that parent 1's genotype should be big G, big R, little r. And that would mean, obviously, we have a heterozygous genotype, meaning that we should have one copy of the dominant allele for each gene and one copy of the recessive allele for each gene. So giving us a nice mixed genotype here. What I'm going to ask you to do now is to apply what we talked about yesterday and to think what gametes can parent 1 make. So based on this genotype here, based on their genotype, what gametes can they make? Remember, we talked about when we were forming gametes, we're going to use that FOIL method. So first outside, inside, last. Go ahead and apply that FOIL method to this genotype and figure out what gametes parent 1 can make. Go ahead and pause the video. All right, so hopefully you came to the conclusion when we do our first outside, inside, last. Parent 1's actually going to be able to make 4 different gametes. They're going to be able to make a big G, big R gamete, a big G, little r gamete, a little g, big R gamete, and a little g, little r gamete. So you hopefully should have come up with these 4 possible gametes for parent 1. To quickly consider parent 2 is also going to be heterozygous for seed color and heterozygous for seed shape. So hopefully it shouldn't be too difficult for us to do the same process with parent 2. So quickly go ahead and consider, what is the genotype of parent 2? I'll give you a second to do that. All right, if we notice, the information here for parent 2 looks exactly the same for parent 1. So when we have this similar information here, we really don't need to do too much work. We've already done the hard work up here. So our genotype for parent 2 is also going to be big G, big R, little r. And with the next question coming up, what gametes can parent 2 make, we should also hopefully recognize that when we have this same genotype here, we have the same genotype for parent 1 and parent 2, they're going to be able to make the same gametes. So it shouldn't be too difficult for us to see that parent 2 is also going to be able to make those 4 different gametes. Big G, big R, big G, little g, big R, and little g, little r. So this is kind of all of the setup work that we need to do. So now that we've considered our parent genotypes, we've thought about the gametes that they can each make, we can now apply this to this nice dihybrid cross here. So this is what I'm going to ask you to first draw in your notebooks. You're going to want to allow a good amount of space in your notebooks to draw this larger Punnett square. I know that the proportions may be a little bit off here on the screen, but go ahead and try to draw as even as a square as you can. It should probably take up at least a third of your page, if not half of a page in your notebook. So go ahead and take a second if you need to flip to a new page and make sure you have enough room to draw. You do want a good amount of space in all of these boxes. So you're first going to start off by drawing a larger box on the outside here. So just one large box, just like how we started off for the monohybrid cross, just one large square. And then very similarly to the monohybrid cross, what I would suggest doing is splitting this box in half, both vertically and horizontally. So we're going to draw these 2 lines next. So you should just have 4 large boxes when you draw these 2 lines. So go ahead and draw those lines, and we should have a large square broken up into 4 large boxes. We don't quite have all the lines drawn in yet. What we're going to do now is basically apply that same idea and consider each of these little boxes as their own kind of monohybrid cross, if you will. So you can even go all the way down here. We're going to kind of break it up into quarters now, and we're going to quickly just draw this line here, draw two more of these vertical lines, and then we're also going to draw two horizontal lines here as well. And if you do that, what you should have by the end of this is a nice big square that hopefully should have 16 hopefully equally sized boxes. So I'll give you a second if you need to pause now. Go ahead and finish drawing that before we go on. So hopefully we should have our nice big box with 16 individual squares in it now. What I'm going to show now is the information we figured out from the last slide. So parent one's gametes, we figured out to be those four. Parent two is going to have those same four possible gametes. So what we need to do now is we're going to try to fill in this information to the Punnett square to predict the outcomes of this cross. So what we're basically going to do now, very similarly to a monohybrid cross, we're going to take these possible gametes, and because we have four different gametes, each of these gametes are going to fall into our regular spots here that we're familiar putting information in for our monohybrid cross. So that's exactly what we're going to do now. So we'll start here on this top line with parent one and start filling in these possible gametes. So we're going to put this big G, big R in our first spot here. In our next spot, we'll put big G, little r. Our next spot, we'll put little g, big R. And then in our last spot, we put little g, little r. So we have properly set up our parent one gametes here by putting each one of our gametes into their own line. And then we are going to do the very same thing with parent two. It's nice for us because in this situation, parent one and parent two have the same gametes. You're going to do the same setup, just going down instead of across. So for parent two, big G, big R goes on this first kind of spot here. Our next spot is big G, little r. Our next spot is little g, big R. And then to finish things off, we do have that little g, little r at the bottom there. So this is our setup for our dihybrid cross, our dihybrid Punnett square. What I'm going to challenge you all to do now is I'm going to actually ask you to try to fill in the rest of this Punnett square on your own. We'll maybe go through a few examples before I get you started, but this is going to be the same process that we've taken to fill in monohybrid crosses. So, for example, when we're thinking about what goes into this box, we should hopefully remember that we're going to take the one from above it and the one from the left. So in this box, we should have these two gametes combining. So big G, big R, and big G, big R combining. When we write this out, we are going to group the letters together. So we're going to keep the G's together, and we're going to keep the R's together. So this is what happens when we combine these two gametes. We have our two big G's and our two big R's. We're now going to consider this box. So if we look again, we're going to take the gamete from the top, and we're going to take the gamete from the side and distribute it across here. So hopefully we should see when we combine these two gametes, we should get something slightly different in this box. We should get big G again, but we have a little r here. So we take our big R from here and combine that with a little r here. Our genotype is slightly different between these two boxes. So there is a bit of need to pay attention to detail here. Let's try another one before I let you go on the rest of the boxes. So in this next box here, again, we're going to start from the top and bring this one down and combine it with this gamete, bring it all the way across to this third box. And what we should hopefully see when we combine them is going to be big G, big R, big R. So that's going to be our combination here. So I'm going to ask you now to do your best to fill in the rest of these 13 squares in our box here. So it will take you a while, but do make sure that you're taking your time. You're thinking about how these are going to combine together, and you're paying attention to when we have big letters, when we have little letters, and that we're grouping them and doing our best to put the big letters first if they are there, and we're grouping by G's and R's. So go ahead and pause the video, give this a shot. All right. So hopefully you were able to attempt to fill in these Punnett squares. If you didn't get it right this first time, that is totally okay. This is a pretty tricky thing to do. So let's quickly go through and see how our gametes combine in the rest of these boxes. So our next box here is going to be a mixed one. It's going to be big G, big R, little r. And then our next one, we'll just quickly scroll through these. And that should be our completed dihybrid Punnett square. You can go ahead and take a second, pause the video if you need to. Go ahead and compare the Punnett square being shown right now to what you ended up getting on your Punnett square. So you hopefully were able to maybe mend any mistakes that you might have made when you were putting this in. Again, it's totally okay if you even messed up quite a few of these boxes. We're still learning, and we'll definitely practice this more to make sure we feel comfortable with these types of crosses. So now that we've filled out our possible offspring, hopefully you remember that when we make a Punnett square, each of these boxes represents a possible offspring in this cross. So we have created these possible genotypes, but very similarly to our monohybrid cross problems, now that we have the genotypes, we now need to consider what are the phenotypes of each of these offspring going to be. So again, as a reminder, we have our alleles up here. When we have a big G, that's going to code for green seeds. When we have a little g, that's yellow seeds. Big R is round seeds, and little r is wrinkled seeds. So we're now going to do a slightly complicated phenotypic kind of analysis of our Punnett square here. But I'm going to ask you to write down near to your Punnett square is this information here. So what we're going to try to figure out when we're wrapping up this problem is we're going to try to figure out the number of each offspring that fit into these different phenotypic groupings. So what are the number of offspring that have green seeds and round seeds? What are the number of offspring that have green seeds and wrinkled seeds? Number of offspring that have yellow seeds and round seeds? And the number of offspring that have yellow seeds and wrinkled seeds. What you're going to need to do is basically go one by one through your dihybrid Punnett square and basically think, if this is the genotype of my offspring, what phenotype will they show? So if they have big G, big R, we'll do this one as an example together. If we think back to the rule of dominance, if you have at least one copy of a dominant allele, you will show the dominant trait. We hopefully recognize that this offspring here has two copies of the dominant allele for both traits, so they're going to show the dominant traits for both genes. So this first offspring is going to be green and round. What you could do to keep track of this is you could either do tally marks by this and then add them up at the end, or you can make a little marking in the box saying that this is G/R to know that that offspring is green and round. Let's quickly maybe hop over to this box as an example. Yeah, we'll go to this box as an example. So if we take a quick look at their genotype, they have big G, little r, little r. So if we have that genotype, again, we need to consider the principle of dominance. What phenotype will they show? So if we look at our G's here, they do have one copy of the dominant allele, which means they should show the dominant trait, which is having green seeds. However, here we see that this offspring has two copies of the recessive allele for the seed shape gene, which means they are going to show the recessive trait. In this case, they are going to have wrinkled seeds. So our offspring here has green seeds and wrinkled seeds, which is going to mean that we're going to put a tally mark here for our green and wrinkled category. What I'm going to ask you to do now is to go through the rest of this Punnett square and figure out which of these phenotypic groups is each offspring going to fall into, and then total up how many are we going to have in each of these categories. So go ahead, pause the video, and take a second to do that. All right, so I know this can definitely be tricky, but we'll see how we did. When we count these all up, what we should find from our phenotypic ratios is that we are going to end up with 9 in the green and round category, 3 in the green and wrinkled category, 3 in the yellow and round category, and 1 in the yellow and wrinkled category. So hopefully we can quick point out where we would find these. So all these across the edges here are going to be in this green and round, that first category. And then we can also draw a line here and see that if we go straight across here, these are going to be green and round as well. So these two in the middle. And then let's look for a green and wrinkled. So that's going to be this one here, and this one here. And then our yellow and round, that is going to be these 3 right here. And then we are going to look for our yellow and wrinkled, which is going to be in our bottom corner here, just our lonely little yellow and wrinkled seed. So that is how all of this should have played out. That's how we should analyze this Punnett square. How I want to wrap up this video is by drawing some general trends of heterozygous genetic crosses. If we hopefully remember, when we have a heterozygous genotype, that means that we have a parent that has one copy of each allele, one dominant allele, one recessive allele. So if we think back, if we are going to think about the general phenotypic ratio of monohybrid crosses of 2 heterozygous parents. So I know that's a mouthful, but if we have 2 heterozygous parents, they have one dominant allele and one recessive allele, what is going to be their phenotypic ratio? How many of their offspring are going to show the dominant trait, and how many are going to show the recessive trait? I might suggest to you maybe drawing out a little mini Punnett square here, a little tiny monohybrid Punnett square to quick figure this out. If we have 2 heterozygous parents, how many of the offspring show the dominant trait? How many show the recessive? Go ahead, take a pause and try to figure that out. What we should see, if we did this correctly, is that in this case, 3 of our offspring will show the dominant trait and 1 will show the recessive trait. So every single time we do a monohybrid cross with 2 heterozygous parents, this is the phenotypic ratio that we will see every single time. Now let's consider the same thing of a dihybrid cross of 2 heterozygous parents. So if we have 2 parents and they're both heterozygous for both genes, what is the phenotypic ratio of our offspring going to be? To consider this, I would definitely suggest you thinking about the example we just worked on. Go ahead and take a second. What is their ratio going to be? If you were to take a look back to that example that we just did, we should hopefully see that 9 of our offspring are going to be dominant for both genes. 3 of our offspring are dominant for the first gene in the pairing and recessive for the second gene. 3 of our offspring are going to be recessive for the first gene in our pairing and dominant for the second gene. And then lastly, we are going to have 1 recessive for both genes. And again, when we're thinking about these ratios, these are just general probabilities. So we can say that 75% of the time, 3 out of our 4 offspring are going to have this dominant trait here, and 1 of them is going to have the recessive trait. 9 out of 16 are going to be dominant for both genes. 3 out of 16 are going to be dominant for the first gene, recessive for the second gene, and 3 of our 16 are going to be recessive for the first gene, dominant for the second gene, and then 1 out of our 16 offspring are probably going to be recessive for both genes. So I hope that this kind of wraps up dihybrid crosses for us. We will be practicing some of this more in class, but thank you all for watching.